In this video, we're going to talk about enzyme inhibitors, okay? So there are certain molecules that can block enzymes working, and that's basically what we're going to talk about here. We're going to talk about the two different types of enzyme inhibitors that exist, and then we're going to talk about how you can distinguish different types of inhibitors using graphs, okay? And that'll hopefully make sense in a couple of minutes. So, enzyme inhibitors can be competitive or non-competitive. These are the two different types of enzyme inhibitors. So an enzyme inhibitor is something that blocks an enzyme from working. A competitive inhibitor is one that blocks an enzyme by inhibiting the active site. Okay, so that means that in a normal chemical reaction, an enzyme is going to be taking in its substrate in its active site, right? That's the portion that it binds to. And this active site has a specific shape that matches the substrate, right? That's what we call the lock and key mechanism. And what a competitive enzyme inhibitor does, the reason why we call it competitive, is because these molecules have a similar shape to the substrate and can therefore bind to this active site. Okay, so they are competing for the same active site. And the more there are of these competitive inhibitors, the lower the chance that the substrate is going to work. So a competitive inhibitor is going to decrease the rate of functioning of an enzyme, right? That makes sense because if it's blocking the active site, then the substrate can't get access to it. The other type of inhibitor is what we call a non-competitive inhibitor. Now, these inhibitors don't bind to the active site, but instead they bind to another site on the enzyme. Okay, so remember, enzymes are proteins, and these non-competitive inhibitors will bind to another region of the protein. And this is what we call the allosteric site. And when it binds to the allosteric site, the active site changes shape. Active site changes shape. And why does that influence the rate of enzyme activity? Well, that means that the substrate can't bind to that active site anymore, right? Because it's just not going to shape that matches. And therefore, you're going to decrease the rate of enzyme activity, which means you're inhibiting it. So you have to be able to distinguish different types of inhibitors from each other by looking at graphs where you have a substrate concentration and a reaction rate on the y-axis. Okay, so let's just explain what this graph is showing. What this graph is showing is, so each of these three lines represents a different beaker. In the first beaker, in the blue one, you have a certain amount of enzymes. Okay, let's just imagine there's two enzymes. And then what they are trying to do is that they're trying to convert these red molecules into green molecules. Okay, so that's what they're trying to do. Completely generic example. And as we move to the right along this x-axis, what you're doing is that you're increasing the number of red particles that you have, or the number of substrates that you have in the same volume, which is what we call concentration, right? So you're increasing the concentration. And this is going to increase the reaction rate, which makes sense, right? Because these active sites, they need to bind to the substrate in order to work. And therefore, if you have more substrate, there's a higher chance that the active site will be able to bind to it. And therefore, it's going to increase the rate at which the enzymes are working. Okay, so as you increase substrate concentration, you're going to increase the rate of activity until you eventually reach a plateau, right? And this plateau is reached because the active sites are working at their maximum rate. As soon as it gets rid of one substrate because it forms a product, there's already another substrate taking its place, okay? And therefore, when you're at this point where they're working at maximum efficiency, increasing substrate concentration won't matter anymore because you're already reaching this V maximum rate. Okay, so that's the first graph. And then in the second beaker, which we're going to draw in blue here, you have the same conditions. Okay, so you have two enzymes, okay, and you have this substrate, and then you're increasing the substrate concentration over time. But within this beaker, what you also have is a competitive inhibitor. Okay, you have a competitive inhibitor which is competing for those active sites. Okay, and what that's going to mean is that at certain concentrations, right? So if you're at this substrate concentration here, at this point here, you can see that the rate of uninhibited enzymes is higher than the inhibited enzymes, right? Which makes sense because when there's these substrates, that are competing with the inhibitor, well then when the substrate concentration isn't very high, well then those inhibitors are going to bind maybe instead of some of those substrates. So it decreases the rate at which the enzymes work. But as you can see, as we increase substrate concentration, eventually we reach this point of Vmax. Now, why is this? Well, imagine that we just had two enzymes, right, as we do here, and we also had two competitive inhibitors in present, okay? Now, there's a chance that these competitive inhibitors will bind to the active site instead of the substrate in this point, right? Because there's two competitive inhibitors, and then 1, 2, 3, 4, 5, 6, 7, 8, no, 1, 2, 3, 4, 5, 6, yeah, seven different substrates. Now, so that means you have a 2 in 7 chance of binding to, or 2 in 9 chance of the inhibitor binding instead of the substrate, right? But as you increase substrate concentration, what you're doing is that you're increasing the number of substrates in this liquid, right? And what that will mean is that eventually the chance of a competitive inhibitor binding to the active site is much smaller, basically so much smaller than the chance of the substrate binding, that effectively it's like the inhibitor wasn't there at all. Okay, so that's why you still reach this same plateau, this Vmax eventually. It's just that you have a flatter curve. And then finally, we have this third curve down here, the green curve. And in this beaker, you can probably guess what's happening. You have the same conditions again. You have some enzymes. You have some substrates that are trying to be converted. But then you also have some non-competitive inhibitors, right? And they're binding to the allosteric sites. Now here, what's going to happen is that you're going to block the rate of enzyme activity, right? For the same reason as a substrate, you're blocking you're changing the shape of the active site, right? Which means that the enzyme can't work properly. But why is it that we don't reach this same Vmax, this same rate as we did for a competitive inhibitor? Well, as you increase the substrate concentration, right, the substrate and the non-competitive inhibitor are not competing for the same active site, right? So it doesn't really matter that these enzymes are being outcompeted, or there's more of the substrate, because they're not competing for the same site. And so as substrate concentration increases, initially you're going to have an increasing rate, right? Because it just means that there's a higher chance that a substrate will bind to an active site before the allosteric site gets to it, right? But at a certain point, you're going to reach this Vmax, this maximum rate of working, but there's going to be a certain number of enzymes that have these allosteric inhibitors bound to them, right? And so they're not going to bind a new substrate until this non-competitive inhibitor kind of chooses by itself to let go. But that is kind of independent of the substrate concentration. So as you reach this plateau, you still reach a flattening, but it's just at a much lower rate than with a competitive inhibitor, because the substrate can't outcompete the allosteric inhibitor. So what are the key points to take from this video? The first is that inhibitors are going to prevent enzymes from working normally. There are those two types of inhibitors, the competitive ones and the non-competitive ones, and that you have to be able to deduce which is which from graphs which show the relationship between substrate concentration and enzyme rates. So I hope that made sense.